there's a lot of other really cool stuff that she wanted to show. That's how much different stuff she did.
Mysterious.
So Silvia has a really, I think, a really interesting background. It's not, I think, I know. It's very interesting. She came from Romania. In Romania, it was a very difficult time, so she went through things that none of us could really experience. I mean, there, I would say pepper spray would be a form of aromatherapy. So anyways, but then she came over here with her husband, and she got into the nursing profession, and then her husband tragically died very young, and instead of kind of, you know, dealing with it and just trying to like go on, which would have been fine, she actually did something really remarkable. She got a Bachelor of Science and then a Master's from Sacramento State University, and then started the PhD program here in the biochemistry, molecular and cellular and developmental biology. And she was also part of the biotechnology, designated emphasis in biotechnology. So she had a lot, and she was really, especially the first couple of years, you were really engaged in a lot of things, but eventually, she actually had 2 different fellowships that she was awarded, which was really nice. She did a internship at Novozymes as well, so there's a lot of things interspersed in here, and she worked with a lot, and the main thing is you got to know a lot of people, a lot of the faculty members here on a very close basis, which is really remarkable, and I'm really happy about that. So she'll show a list of names, and these are people that just didn't consult, but she could email them and call them, and they would talk, and they treated her as a colleague, and it was really great to see. So I'm thinking if I forgot anything I wanted to say, but yeah, I'll turn it over to you.
She was one of our NIH biotech fellows.
Right, exactly. That was one of the fellowships exactly that I was talking about there. The training grant is really, it's a nice thing. And so she was engaged in a lot of different things and made a lot of people, you know, I think days better having talked to Silvia. So take it over, Silvia.
Thank you very much for the nice introduction. It's been an extraordinary journey. I started about 7 years ago. I never thought it could have happened. It started with me looking for a hobby that would not take me away from home, as you said. And it's a weird hobby to go and get your bachelor's degree, but it worked out. And it's even weirder to get your PhD degree, but it worked out also. And it's been absolutely no.
Oh, you want that? I'm sorry.
Silvia, yell at me. Don't touch the lights.
How's that one? Is that okay?
Yeah, it still works.
Okay.
Maybe a little more.
A little bit higher? Okay.
Thank you.
Okay.
So what I'm about to present to you, to me, is extraordinarily exciting. It's fun. It was fun to do it. It was fun when it worked. It was even fun when it didn't work, and it was even more fun when we got the results that we got. I'm a fencer. I was a fencer, a performance fencer. So the title of my presentation is "Spin Label Fluorine Compounds Are a Versatile Sword in the Fight Against Amyloid Beta Peptide of Alzheimer's Disease," and that's how I imagine it. It's a nano-molecular sword that cuts away the amyloid beta, and I really like it. So let's go on. This presentation, as John mentioned, I have a lot of results that this presentation got cut really short by a good number of slides before we got here. That possibly physically I cannot present all the results that I have. So we focused on 3 main aspects on the fight against Alzheimer's disease. The first one is, so basically we have a compound. It's a spin label fluorine compound that has a nitroxide label on it that is a very versatile compound. So the first aspect that we're going to focus on is has to do with the intracellular amyloid beta and the protective role that the spin label fluorine compounds have against it. The second aspect will be the role that the spin label fluorine play in the amyloid beta oligomerization. And the third area of focus will be how can we image the amyloid beta, and I'll get in details on all those things. All those three aspects of what we have been doing and will hopefully continue to do are very important in trying to devise a strategy to conquer, at least, try to figure out what's going on and why it's going on. And at the end, we have a couple of future directions that we're going to talk about it. Why Alzheimer's disease? Besides what I'm putting on this slide, I was touched many, many years ago when a patient of mine that had, by the way, I work in cardiac surgery ICU, and I see a lot of people. And this patient also had Alzheimer's disease, and he was a physician. And he ended up being on national TV. And I didn't know what he was doing because he kept asking for a camera, a video camera, and his family brought it. He was taking a video of himself, telling his wife and his daughter how much he loved them. And he said, I want to tell you every day how much I love you, for as much as I still understand what that means. And it's a you don't see it on the outside, but it robs people what I think of the most precious thing, their memories. And that is, on many levels, a lot worse than probably any other disease. But also, it's very widespread. As you see here, there are 5 million Americans currently estimating to go, about 5 times more by 2050. The most important thing, it's a tremendous economic burden, not only on the society, on the healthcare industry, but on the healthcare caregivers. The life expectancy has increased. The families have to leave work to take care of their parents or their siblings, and it's really, really a devastating effect. Briefly, Alzheimer's disease is a very lengthy, very complex process. The most important part of this is that there are two pathologies for it. One that has to do with insoluble extracellular amyloid beta and the intracellular neurofibrillary tangles that result from the aggregation. There are many lengths of the amyloid beta, and I'll get into details later, but the most common are the 40 and 42 amino acid long amyloid beta. And the greatest problem that it is that so far the diagnosis cannot be made for certainty unless it's a biopsy, post-mortem biopsy. And also, the pathology of Alzheimer's disease starts way before we get to even the clinical diagnosis of dementia. So if we could zero in and try to focus on what ways to diagnose early Alzheimer's disease, that is a great gain for community. I'm presenting here with a very brief, I'm not going to go into large details, but just so you see that it's a very broad pathology that develops. The amyloid beta is as I said, 40 to 42 amino acids peptide that it's produced from proteolytic cleavage of amyloid precursor protein by the beta and gamma secretases, then is transported outside the cell by transporter like apolipoproteins. In the process, if there is a defect in the transport, it can oligomerize just by increasing in concentration and lead to amyloid plaque formation. The presence of the oligomers lead to toxicity, dysfunction, to toxic effect of the amyloid plaque. They disrupt the synapses, they inhibit the synapses, they lead to inflammatory processes. The trafficking inside the cell also plays a major role because it can lead to radical oxide species production. Not only the lysosomal degradation, but currently there's research out there underlying the role of exosomes, early exosomes production. Our journey started a little bit before I got into this lab with a known fluorine binding compound. This fluorine molecule that you see here in the Leeway Gene Lab that was developed, it was known that it has a affinity for amyloid beta. And it was known that it limits the A beta aggregation by intercalating between the beta pleated sheet of the A beta. However, when Voss lab developed the spin label fluorine by adding a nitroxide moiety to this compound, as we say here in red, something very exciting happened. If you look here, when this compound is just a fluorine compound, this is a cell viability assay. The red line right here represents cells that are grown in the presence of tetracycline. And I have to make a quick parenthesis. Those cells are neuroblastoma cell lines that are engineered to produce APP as soon as the tetracycline is removed from the cell culture. And coming back here in the cells that tetracycline has been removed, the survival of the cells is very low. When we culture the cells with the spin label fluorine compound, you can see that the survival rate has shifted. The same right here. So the red line is just, again, this one, high survival. And after a couple of days, even if they have tetracycline, the nutrients, get exhausted in it. So, but the very exciting thing is that this black line here has shifted tremendously. So from here, basically from here, it's really high here. So that was the beginning of the spin label fluorine journey that we engaged on. So the first aspect of our research has to do with intracellular A-beta toxicity, which we're focused on evaluating the spin label compounds as bifunctional therapeutic agents, and that has to do with the two moieties, the fluorine binding moiety and the nitroxide spin moiety. And especially in the view of the fact that more and more there are reports in the literature that the most soluble oligomers are actually responsible for the toxicity. There is some sort of disconnect between the plaque pathology and the degree of disease, and where there's not as much plaque, amyloid plaque, but the disease is really advanced. So the notion is that it's probably the soluble A-beta oligomers that play a big role in this. And it goes both ways. It's the soluble oligomers produce neurotoxic damage on the neurons, increase oxidative stress and produce reactive oxygen species, and increase oxidative reactive oxygen species leads to the increase in A-beta production. So it's a circle, it's a vicious circle that it's hard to get out of it. We know that we talk about, to verify that we talk about intracellular amyloid beta, we did a really cool experiment that I'm really proud of it. This is probably, am I right to say that it's the first time that this kind of picture was taken? Not that I'm, I do want to be modest, but it's really a cool picture. So we took a super resolution structure illumination scene image of FSB stain. I'll explain what those things mean. So basically those are human neuroblastoma cells. They're called MC65 that overexpress APP protein in the absence of tetracycline. And when we remove when they're grown in tetracycline, you see a nice neuron body, nuclear membrane, nice and round and smooth and all looking great. When we remove the tetracycline, 12 to 24 hours, you see those inclusions that are stained. This FSB has a very long name, but is a known amyloid beta binder. It binds, it's a fluorine styrene based dye that binds to amyloid. And those green areas here are amyloid deposits. And you see how it just absolutely encroaches on the nuclear envelope and produces damage. And it's quite actually, we don't have the image, but for older cells that stay without tetracycline longer, basically the whole cell turned green. when we added the spin label fluorine compound to it, removed the tetracycline and added the spin label compound to it, you can clearly see that there's still a little bit of encroaching on the nuclear envelope, but the amount of A beta is greatly diminished. So we're really pretty excited about being able to image this. So much so that now that we know it's intracellular, or we sort of have approved that it's not intracellular, we went further and explore the other possibility that the spin label fluorine offers us, which is radical oxide scavenging. As I mentioned, the nitroxide moiety of it having a single electron on it is able to scavenge and just trap the radical oxides. And this is a confocal image of same cells, MC65 human neuroblastoma cells that are grown in the absence of tetracycline and SLF, and in the absence of tetracycline plus our compound. And we used to image, we use a cell rocks deep red, which images radical oxide oxidative damage, and you can clearly see the red areas around the nucleus, and the nucleus is stained with that. The red areas, the punctate red areas around the nucleus are really clearly abundant, which compared to the ones that are grown in the presence of SLF, they're actually almost diminished. As a matter of fact, as a parenthesis, I was working yesterday on the Raman spectroscopy, and I reconstituted the A beta 1 to 42 in ACN that has no SLF and ACN with SLF, and I actually could see it by naked eye how the one that had SLF in it completely dissolved and didn't aggregate, and the one with without SLF had it. So that was pretty cool. So having said that, we're pretty excited that somehow we made inroads on this pathway towards trying to understand what happens in A beta aggregation, oligomerization, and the additional processes that are implicated in it. This is a schematic representation of what we think happened. A beta starts as A beta, it oligomerizes. Those are native small oligomers that, if left untreated, progress to fibrils. Those are still disorganized, but then you see that fibrils are really, really organized. If anybody remembers the folding funnel from the biochemistry class, they are now in the stage where they cannot be refolded right here. Adding our this little green squiggly, it's our representation of our SLF compound. We actually used this pathway to do a couple more experiments that I will be presenting in the next slide, in which we can, we're investigating the effect that this SLF has on the small oligomers, or we call them monomers. You just have to understand that A beta is a highly intrinsically disordered peptide. So at any given time, there are some forms of each of them, monomers, oligomers, dimers. We did some experiments to try to optimize, and we have a low aggregation buffer that keeps the monomers in the monomeric states, and a high aggregation buffer. But interesting, the pathways will meet at some point, because if we add the SLF to the monomers, we think that there is, the SLF is a highly hydrophobic compound. So one of the theories that we're working on is that upon binding to the A beta, it produces a small conformational change. A beta tries to protect the SLF from the aqueous environment, and this new conformation will still aggregate, but it will be a more compact formation, as opposed to SLF that is added to already oligomerized, bigger oligomers, and it will be, this one will result in a larger, less compact formation. However, this one can also, if the conditions are right, there's enough SLF and conditions are right, this one also can progress towards breaking of the more structured monomers out of the oligomer. Our previous studies done by Robin Altman here show additional supporting evidence through EPR spectroscopy and fluorescence correlation spectroscopy that this pathway is very plausible and things might happen that way in the cell. So having said and presented you with all those options, we said, okay, so SLF affects A-beta oligomerization, but how about the effect on A-beta conformation that I was talking in the lower part of the slide? So this is another thing that I'm super excited about. It's called a circular dichroism spectroscopy. We acquired this a couple of years ago, 3 years ago maybe, and I got sole possession and use of it, which is really, really cool. So circular dichroism spectroscopy, briefly I'll describe it. So amino acids have a chiral center in their protein, in their amino acids have a chiral center. So CD spectroscopy looks at polarized light in one single direction, and it looks at it in 360 degrees. I was trying to get the movie that it shows how at each point of rotation, when it polarizes and it rotates on 360 degrees, the software constructs a vector that is the resultant vector at that position, and then it moves to the other vector. So it was really, really cool. So anyway, briefly, one can determine the secondary structure of protein by looking at CD spectroscopy in the far UV spectral regions. And the difference, you have to have a chiral center because the instrument looks at the differences between the left and right polarized light. Otherwise, you can't tell the difference. So then, based on that, you get characteristic CD spectra for each type of secondary structure. I have to emphasize that this is a qualitative measurement, it's not a quantitative measurement, or at least it's a very hard quantitative measurement as we figured that out. So most characteristics, the red trace shows spectral characteristic for alpha structure, blue for beta sheet, and black for random coil. So they will not look exactly like that, but very close to this. So once you see the next slide, we can tell closely enough that we're able to identify those specific secondary structures. As I mentioned before, we're looking at the major, the effect that SLF has on the secondary structure of amyloid beta. And upon the experiment, the conclusion is that there's no huge major changes, but it slightly reduces the amount of beta structure and disorder structure. So this is, in the left side of the figure in the left panel is amyloid beta that we incubated and analyzed through CD spectroscopy in the absence of SLF. And you see the beta structure here in the red, and the alpha in the black, like really, really low. In the right panel, we have incubated A beta with SLF, and the differences are not great, but we can tell that if you look here, the beta and alpha, the beta turn and the unstructured have flipped right here, with the unstructured being pretty much the same, but the beta turn has decreased, the beta sheet structure has decreased. Alpha slightly increased, but not tremendously. But the alpha and beta have flipped, so that's encouraging. Again, as I said, this is a qualitative assessment, so not measurement, not a quantitative one. So in order to try to quantify and characterize the binding of SLF to the A beta, we went and employed a quite interesting technique it's called surface plasma resonance measurement, and those experiments showed us that amyloid beta, when SLF alters amyloid beta oligomerization as a modulator of the peptide aggregation and the toxicity. And this is a quite extensive work that we've done in collaboration with some scientists from Finland. So not all the experiments that you see here I've done myself, but I've participated in the analysis, and it was quite fascinating to do it. I was really fascinated. So surface plasma resonance is what is done with an instrument that most people know probably as Biocore. And Biocore, the principle of this one is that at the surface of the gold chip, there's a gold chip right here, and the plasmons here flow in a nice undisturbed pattern until the ligand and the analyte bind together and causes a disruption in that flow, and you get a signal. Basically, I mean, I could go into more details, but and this is an incident light, and it's refracted, and there is a shadow here area that tells the intensity drops upon refraction, so the angle shifts. This angle here shifts. The multi-parametric surface plasma resonance, the difference is that it uses the same gold sensor, but it uses 2 lasers, 2 fluidic chambers. So you get like, I don't know if I could say that, it's almost like a 3D, characterization of the changes that happen on the gold chip, on the chip. So we devised an experiment just so understand the schematics a little better. This is the gold, this is the glass. We have the lasers going in. We have the electrode, which is gold. We put a tile self-assembly layer. There are many ways to do that. We can bind the A-beta on the surface through antibodies. We chose to bind it to a biotinylated biotin-streptavidin compound because we thought that this provides less interaction and affects less results. And we had the biotinylated A-beta, which is represented here, and the analyte, which is this time is the red squiggly thing. And when we did the measurement, so it was really easy. We reconstituted the A-beta plus minus SLF, injected it in the instrument, and measured. So as it binds to the self-assembly layer, you see an increase in the concentration. Then you wash it, and then it decreases, and then you inject another a higher concentration of SLF, and then you wash it again, and increase so on. So what is interesting in the experiment is to watch how the baseline changes with time. So this distance between the baseline here and the last one is how much of the SLF is retained, bound basically to the A-beta that is bound to the SAM layer. So that is how we measure. And then we did additional measurement. We can measure the layer thickness that is there. We can measure the refraction index of the surface. So that it's only complementary measurements that tells us that indeed the SLF binds to the A-beta. We use a special software that was provided by the company that we had the instrument from, and that calculated the binding kinetics of the A-beta. And in conclusions of the binding is that SLF binds both the biotinylated and the non-biotinylated. So to come back to SLF, we know bind the biotinylated. We got a very strong affinity constant, but where we wanted to see it, does it bind the non-biotinylated A-beta? So we flown some non-biotinylated A-beta over the biotinylated one. Some dimerization probably happened, and then we flown the SLF, and we got the same result. It looks exactly like this. So then the analysis that fits that we did were more appropriate for the 1 to 2 binding model. So conclusions was that A beta provides 2 binding sites for SLF in the monomeric dimeric state, one with strong affinity, the other one with a weak affinity. When we went and checked how SLF binds to the oligomeric A beta, we again split the experiment in two. We took the A beta monomers, let them incubate and oligomerize, one in the presence of SLF, the other one in the absence of SLF. And when we did the measurements, we fit also the kinetic calculations so that there's still there's two binding sites, but the difference in them is not so great. So there's some interesting speculations we could do about that. Basically, we know that now that SLF inhibits the self-association of A beta, if we go back to this slide here, whether when SLF binds to oligomeric A beta, the complex is very large, and somehow because of the conformational change, the binding sites with lower affinity are the only one exposed, or the conformational change affects the binding affinity such that it decreases the stronger binding affinity. I mean, a lot of other experiments will be necessary to elaborate on that one. The other interesting that I like to keep it here is that we saw that when the oligomers pre-incubated in the presence of SLF, the baseline, if you remember what I was showing you, so keep in mind, look at this picture, like difference between this one and this one. The baseline of A beta oligomers incubated in the presence of SLF, this baseline, the difference, it was much larger when the A beta oligomers were incubated in the absence of SLF. So the oligomer size was larger. So we also postulated that probably A beta not only that prevents the oligomerization, but leads to a more compact structures. So the additional measurements that we've done, really cool. We measured the layer thickness, which I was really excited about that one. So this instrument is so sensitive that it can actually measure how much A beta deposited on the surface. So it was again, remember the schematic that I showed you was 32 nanometers versus 1.5 nanometer. So this was in the absence of SLF, this one was in the presence of SLF. It was correlated by the surface mass density increase that calculates how much mass actually, the density of the mass that is on the surface. And this part, I don't know much about it, but it really has really cool pictures. If anybody wants to stay over, I will show them. They did molecular docking and molecular simulations. Always wanted to do that, but I never done it. And the conclusions are that probably the binding of SLF to the surface is non-specific. It's depending on the amino acid side chain. And keep in mind, it's a highly hydrophobic molecule, so that that might play a role. That SLF remains at the surface of the A beta, and we have additional experiments with whatever we did after that in the next step, which is equally exciting. It shows that SLF is retained actually in the brain of the mice. I'm just giving up what is going to come next. And again, another theory that it possibly refolds a binding better to shield the hydrophobic SLF molecules, which we already talked about it. So that's very possible. So the last part of my presentation is actually the most exciting one, that has to do with imaging the amyloid plaque in the brain, which, as I said, it will be easier, it will be of high value if we could image early and see what other treatment options, because by the time it's diagnosed, it's too late. I mean, the neurons know how to die in one way, so much plaque formation, it doesn't go anywhere, the damage is done. So we know that AD is characterized by depositions of the amyloid beta. Progression is over a decade, as I said, is really, really heartbreaking because people don't know they have it. And because it's such an important, MRI is cheap, available, and if we could find a contrast agent that it's less toxic or non-toxic, so far it's done with gadolinium-based agents toxicity. I work actually, as a matter of fact, a couple of weeks ago, I have injected some gadolinium in somebody, and I have to sign a waiver as the patient's nurse that I'm absolutely sure that they don't have kidney problems, because otherwise they won't do it. So that's interesting. So as I said, this is really exciting because we provide the first demonstration that a nitroxide-based small molecule produces MRI in the brain specimen with elevated levels of A-beta. So what we started with was, I'm going to go over really quick some principles of eMRI. So the spin label part provides contrast in MRI, and it has to do with the paramagnetic quality of this one that produces a proton relaxation enhancements at the surface of the water, dipole-dipole interaction, and it actually, what we're looking for is a loss of contrast in the image that we see. So we have this highly AD transgenic mouse, 5XFAD, has 5 familiar mutation that has high levels of A-beta, and it's a very useful in studying amyloid-related pathogenesis. The first step that we took, this is another really cool image that I have done using a infrared imaging. This is this slide here. It's from an AD mouse, and this one is from a wild-type mouse. This is with anti-A-beta primary antibodies, and secondary antibodies are infrared antibodies. And you can see those little white dots indicative of the presence of A-beta, and none here. That was done just to show the pattern of distribution of the amyloid plaque, and it was better to do it that way as opposed to simply confocal because it shows I could see the whole slice. So this is the really cool, exciting stuff. We're developing, John is developing a lot of spin label compounds, fluorine, resveratrol, coumarin. All those are known to bind amyloid beta, and they all known to have affinity for amyloid beta, and their nitroxide spin is used as a probe for MRI. This is SLF. This is a less amyloid producing mouse. It's an APP-PS, I think it's an APP-PS1 mouse. But you clearly see, but don't need to do any measurements, how darker this is, how much loss of contrast. Those are in vitro slices. We just baited in SLF, washed them, and embedded them in some agarose, and put them under the MRI scan. This is a resveratrol class. The wild type doesn't even show up here. Clearly a great loss of contrast here. And this is a new coumarin class that what we also did, we have a technique that I'm going to get to it. It's called electron paramagnetic spectroscopy and resonance. I'm sorry, electron paramagnetic resonance spectroscopy. We place the spin label in a magnetic field, and depending on the degrees of freedom that it has at that point, it gives you either a sharp line if it has a lot of degrees of freedom, or a broadened spectrum if it's embedded in a secondary structure or that limits its degrees of freedom. So,
Silvia,
uh-huh.
sorry, if you just go back one.
Yes.
So if you just want to point out that if you do the same control, if you take wild type and you mix it with the SLF or these other compounds,
yes.
you don't get any
Thank you.
contrast.
So this is the wild type. So what I did, I took both of those slices in the same condition for the same amount of time. We just soaked them in SLF, 80 micromolar, and washed them together. And when we put them in MRI, we got no loss of contrast here and loss of contrast here.
And then the EPR spectrum is the same experiment. you don't see any EPR signal from
Yes.
the wild type mouse.
Do we have the slice or it's?
Well, no, but I mean,
No.
you
Yeah,
can't
exactly.
see the wild types in these images, they're so faint.
They're so
But they're
faint,
in
it's-
there, but-
yeah, I can tell you there's someone here and there's
Yeah.
someone here.
So
So
that
yeah,
EPR spectrum is actually EPR spectrum of the slice she's looking at in the MRI.
in this one, I
And
took
that
it out of,
shows, yeah, so it's kind of a very important specificity as well.
yeah. And the wild type is just a flat line, there is no signal from it. So it does not retain. And it retains it, I think we did the EPR spectra like what, 3 weeks after? Yeah, so it stayed in the mouse brain for a long time. We went a step further. This is a one-time experiment. We did one mouse. This is ex vivo. Liwei gratefully, Liwei Jean gratefully gave us a mouse. We injected a tail vein injection, about 3.3 milligrams per kilogram. And after 24 hours, we sacrificed the mouse, got the brain slices, the same coronal, hemicoronal slices, embedded them in 3% agarose. The top one is the wild type, the bottom one is the AD, the same one. I believe this is, technically this is a T2, this is a T2 star, but we can see, as opposed to where the slices were literally embedded in SLF, embedded in it, there was contrast, loss of contrast throughout. Here it's a little bit more localized, which is of great interest to us. We think that this darkening is probably an anatomical structure, but we can see this one repeats the pattern that I showed you initially in the infrared image of the mouse with the little dots throughout, diffusely throughout the brain. This one is a better image. Those are overnight images when you clearly can see the loss of contrast produced by the SLF injected in the mouse and no loss of contrast here. So this is promising. Unfortunately, it's just one mouse, but once you get more, oh, there you go. This is, after we did the imaging, we took the slices out, and we have flat cells in the EPR and did the EPR spectra, and clearly see the white type plus SLF is basically a flat line, while the AD mouse plus SLF has a nice pattern, nice signal. So, in conclusion, we believe that SLF is a reliable MRI contrast that has a great potential as contrast agent for MRI. Not only that, but it's also a therapeutic agent because it prevents the oligomerization. We did additional test of toxicity. We had 4 mice that we kept alive for 14 days after we injected them with SLF, the same amount, and really I haven't seen no signs of any toxicity. They died of for science reasons not related to MRI imaging. For the future, we're exploring additional amyloidophilic compounds with potential, as we show in the previous slide. And one interesting point that we made is that because that nitroxide spin label is prone to being prone to oxidation inside the cells, we want to see if we can develop spin label agents that are resistant to chemical reduction, and some of them have like additional methyl around them somewhat to protect them against the cellular environment. Least but not last, I want to thank a lot of people that have been a part of my journey here. First and foremost to John for being my PI for so many years, for being such calm dude that never loses his cool. He's not a micromanager, but he is amazing. I mean, I've learned so much, and exactly the person that I needed to be able to function in that environment. To Madhu, which is our specialist in the lab right there. He helped me making sure I eat lunch and organizing my desk and my countertop and everything. To the DV people, I met Judy Chelstrom, Dr. Chelstrom, many years ago at Sac State when she asked me, so what are you doing? I was presenting a research, and I said, I don't know. And she said, why don't you apply for a PhD? I don't know, I'm too old. She said, no, you're not. So, thank you very much. As I said, that you made my dream come true when I didn't believe that would be.
I even wrote a letter of recommendation to get you in here.
Yeah. To Marianne, Hunter, and Neil, and the whole DB crew, you guys supported me. You helped me get do all the paperwork to get my NIH training grants, to the other DB fellowship, to all the times when I was lost for all the things that, including today, Marianne. With Marianne.
This is where.
To Robin. Robin has been my PI, my postdoc in the lab when we first met. She helped me rewrite all the protocols from scratch. Listened to me. We laughed, we had lunch, we had dinner. We're almost like a married couple. To Dr. Lee Wei Jin and Izumi Maezawa. Izumi, it's just, I don't know how kind and selfless and helpful she was all the time. When I lost my cell cultures one day and I was crying and she said, don't worry, I give you something. Lee Wei for giving me that mouse, that was great. But he also guided my research, was part of my dissertation committee. Milo, I met Milo on my first day of college when I was lost because I couldn't find my car in the parking place because I parked it there at 6:30 and at 4:30 the parking was. And from that first day until last week when he helped me with the mice and teaching me everything, Milo has always been there and amazing. I mean, it's amazing.
Didn't you know him at Science State?
Yes, first day of college. When she said, don't worry, we'll call your husband, we'll walk you home, we'll find your car.
He was in my class.
Oh, yeah. To Angie Louie she's the help and expertise in MRI. Tang for the long hours. We call him my brains. It was my brains. To Jeff Walton. He's the boss of the MRI facility and every time there to just do those long scans. And least but not least to Matt Clement and Jack for being on my dissertation committee and helping me complete my degree. I have a last sentence, if I'm crying, I said, but this is truly dedicated to the memory of my husband and my parents. So, thank you very, very much. Please.
So I'll make a comment first, is that you were a little bit too modest about the SPR work, because you did all the SPR, it was just the modeling part that you weren't involved in really.
Yeah.
So, and also, so the question about you said you injected wild type mice with some of the SLF?
Mhm.
What happened to those mice and how long did you look at them?
But actually more than for 2 weeks. When I told you there were 2 weeks, but Milo needed them alive for 2 more weeks. So there were 4 weeks. He needed them for his research.
And they seemed okay?
Yeah, they seemed just fine. The male mice were fighting each other then. Yeah, they seemed just absolutely fine. But he was doing he had to sacrifice them anyway because they can't keep him for a long time. I think it's for breeding or purposes, I'm not sure. But it was about time, so I said, okay, we can inject them and see. But they were just absolutely fine. We have to change that. It was 4 weeks, not 14 days. Yes.
So could you summarize again, how does the spin label affect the activity of SLF? Is it SLF, that's the compound before the spin label is added?
So no, the SLF is called a spin label
Okay.
fluorine. So the SLF refers to just this one. So the fluorine is this, the fluorine moiety is this one, and the spin label moiety is this one. We initially had, no, sorry. I will get there in a second. We started with the fluorine compound that, if you do a literature search, this is well known to be, to have a high affinity for the A beta. And in the initial studies that Li Wei Jing lab were doing and John lab were doing, to see how this fluorine compound that has such a high affinity for A beta prevents toxicity, possibly by intercalating between, am I right? That we're looking
Yeah,
at that?
just
Between the
inhibiting
beta
oligomer
sheet, yeah,
formation.
oligomer, preventing oligomer formation. So then John thought that if that is the case, and this one is a well-known A beta binder, then we can use it as a probe by adding another part to it that would give us indication where it is, what happened, based on the environment in which that probe is. So based on the environment, we do something very interesting in the lab. We do site-directed spin labeling. So what we can do, we can put a spin label basically at any position on an amino, on a protein. We mutate the amino acid of, not the amino acid of interest, but in the vicinity of the position of interest with a cysteine, then we make a tau binder with a spin probe. The same principle here. We don't need to mutate an amino acid, but we have this compound that is kind of like the direction teller. It will go to the A beta because it binds. So this portion binds to the A beta. This portion can, if we do EPR on it or MRI, can tell us is that compound in the A beta or in a certain, portion of a tissue, is it there? So this is the binder and this is the probe, the indicator.
I'm trying to understand the two curves. Does it shift the?
Yes.
Shifts it to it's about 8-fold increase by just adding that nitroxide.
Okay.
And so that was the first questions was in actually your two imaging figures kind of show both of the properties.
Yeah, exactly.
Which
So
is-
what happens, this single electron is a radical oxide scavenger. So not only that it improves the cell survival because it binds to the fluorine part, but it also scavenges the radical oxide that happens in the process.
If you were to modify it in a way that didn't have the scavenger but had a similar shaped moiety that you added in the same place, do you think it would have the same properties? Or?
On the radical oxide scavenger, no. This one doesn't have the radical oxide. It will be a diamagnetic.
That's a good, and we have actually versions
Yeah.
of this compound where instead of the nitroxide species, we have just a methyl group, so it's
Yes.
N to CH3, and it behaves protection-wise just like the base parent compound.
Just like this.
Okay.
So we have actually quite a few variants of this, and we've published all of the protection and so forth. This is sort of the one that it has, I think, mostly due to its better solubility than some of the other ones, so it gives us the best protection. And if you go to your next
Mm-hmm.
slide of these two imaging. So if you look at this slide right here, this is one property, which is basically keeping. We don't know if it's just the fact that it prevents the oligomer, or prevents the oligomer and also facilitates the clearance, which it probably does. But the unlabeled version would have the same activity as you see here. She hasn't done the super resolution, but they've done some other types of lower resolution staining. But if she goes to the next slide, the parent compound wouldn't have this activity. And so that's why, and we've done a lot of other studies, and Robin has and so forth too, looking at how the antioxidant, and it's just, the nitroxides are actually very unique antioxidants because of their ability to participate with the cell's endogenous glutathione system, where a lot of scavengers, they'll scavenge a free radical, but then like vitamin E, they become a radical themselves, and then they go and they oxidize something else. These cycle with the cell's innate system, and so you don't end up with other radical end products, and I think that's why these are so effective.
Yeah, and also, we hear possible also that if it's SLF, maybe some radical oxide scavenging that will actually decrease the production of A beta as a result of the inflammation that A beta, as a result of the inflammation resulting from the from the oxidative damage. So remember that I was saying that is like sort of a vicious cycle.
Yeah, and actually, what's nice about these too is you're targeting your antioxidant right to the species that you want, the A beta, which is the generator of these.
So the spin labeled compounds are very specific because we know that either the fluorine moiety or the resveratrol or the coumarin are specific for the A beta. And also the spin label is specific for, the spin label is a specific probe. If it's not there, you don't see the signal. So
Does it bind, when you give it to the mice, does it bind elsewhere in the body? Where does it go?
well, we haven't got there, but if there is A beta, which sometimes there is A beta throughout the body, I mean, there can be amyloid, cardiac amyloid, it can be renal amyloid. Is it possible that that's another option? I actually have seen a patient that had heart surgery and it was a failure to thrive, and when they did the biopsy, he had cardiac amyloid. So they did a conga red stain, like a fast stain in, you know.
Mhm.
So it was interesting. I mean, so the possibilities, I think, are great. Now, I don't know if the amount of amyloid in the heart or kidneys is as much as in the brain to be enough to give a contrast, but that's definitely a possibility to an avenue to look at.
Since you're part of the biotech program, I'm wondering, what's the future of something like this? Is this patentable?
We do have, we have those 3 classes there.
Mhm.
There's a patent that we just pending, just submitted for it. So otherwise you wouldn't be talking about it right now.
Right,
So like,
right,
it was-
right. So you get about 1 year
Yeah,
provision.
yeah, so, we'll see. I mean, it's the first time I've ever done something like that, but it's
The industry, I would think, is very interested in this.
It's actually, it's really, I mean, the penetration, I mean, Silvia had other slides we had to cut, but we have data on brain penetration other than the EPR.
Mhm.
The EPR has actually been really nice because it's like correlated to the exact sample that she's measured, but we do have just some basic, but the main thing is getting funding to do like animal studies, which is a huge project, so hopefully.
Expensive.
Yeah, but there's all those questions, the metabolism, how it's cleared, as like Tom raised, all of those things need to be established, but it was good news that she injected a few mice with quite a bit of it, and it didn't seem to bother them, so they seem to tolerate it
Yeah,
very
we
well.
have, I think a biochemistry with like, what, 0.5 plasma to brain, brain to plasma
Yeah.
retention.
So,
It's exciting.
yeah.
It's
Yeah.
very hydrophobic. Is that a problem in working with it? I mean, it has some solubility in water.
Yeah, I mean, I can use DMSO or ACN. It didn't really bother me for cell cultures or for mice. It didn't seem to bother the mice that injecting DMSO in them.
So yeah, I think it was a 50%
Yeah,
DMSO.
50% DMSO.
The DMSO I was surprised when I read about how well it's tolerated. I know in Russia, a lot of people take it, like
Yeah, take
tablespoons
it,
of it.
yeah.
That's
Or
it.
they.
I used to
And they
rub it
rub
on
it.
my legs when
Yeah.
I
But
read
I told you, I mean, they were like
But
4
it makes it
weeks.
a little bit of acid.
Yeah, I know.
You get-
My wife
get
has
the
an uncle
muscles.
who like rubs
They
it
do
in his
it in
hair
the-
every day.
Well, they do it in the.
Yeah, I'm not kidding you.
Veterinary medicine.
That's what made me look it up. I was like, what? Yeah, I know, they do it right with
Yeah,
it.
that's why we did it.
When we
Yeah.
had that single mouse and I had to inject him, I was like, oh my God.
I know, we're like in the lab with like, you know, goggles and stuff, and it's like uncle like rubbing it all over his face.
And the mouse is gonna die. Well, it didn't. It just did fine. And so did the other 4 mice. And,
Yeah.
And they got, well, I think it was in the 80s that they got really scared of it, you know, but we were using.
Well, we
it in
add
the
DMSO
70s.
to when we do frozen stocks of cell cultures. Yeah, right?
Yeah, you can, it's a cryoprotecting.
Yeah,
use
It's a
all the time,
cryoprotectant.
so.
Yeah, I'm still here.
But it's not, I think, it's probably like 100 micromolar, Tom, in water is about, in buffer is about as high as you can get these compounds.
Yes.
But we can make ones that are more, but it seems like, when we do our in vivo, whether it's cell, or we do cell culture or mice, it's definitely distributing. So there's enough, whether it's probably binding, once you inject it, it probably hops onto things like lipoproteins and BSA, things like that, and then it's taken.
Madu has it dissolved in water, didn't you? But we wanted a higher concentration because we can inject only 100 microliters per injection. So in the mouse. So.
Yeah, we're glad you're still continuing on this journey.
Yeah.
Yeah.
Well, let's thank Silvia one more time.